Well, good morning, everyone. First of all, I want to thank the chairman and the organizers for inviting me to Porto. It's a beautiful city, and I'm sure my friends in Canada and me being here at this time. Today, I'm going to talk about how we use networks to change care, and also I think about what kind of paradigm we should be using, and how should we be thinking about improving quality of care. First of all, I have no conflicts of interest to declare. So I'm going to talk about the Canadian Neonatal Network, the concept that we've been using in Canada called EPIQ to improve the quality of care, the different trials that we've been doing, and then end up with some considerations for us to think about in terms of improving care for the future. So the Canadian Neonatal Network was founded in 1995. It comprises all 30 neonatal intensive care units across Canada. We are a bit different from most of the other networks around the world in that, first of all, we are population-based. Second, we collect data from every baby admitted to any NICU across Canada, so not just very low birth weight infants, but all babies. Third, it's a very high quality database in the sense that we collect not just outcomes, but also processes of care. And then we use the processes of care to improve the practices that we have. And I'll explain a little bit how we do this. We also collect data that is standardized on follow-up up to 3 years of age for all babies admitted to the NICU, on all babies transported to the NICU, and on selected babies who go for surgery in the NICU. So it's a fairly extensive database. Now, in the usual Canadian tradition, we operate with no money. Every hospital pays for their own data collection, and research projects are funded by research grants for specific projects. So why a network? Now, if you look historically, we find that neonatal outcomes actually improved around the world from 1960s to the 1980s, and dramatically. However, from the 1990s onwards, for the next 2 decades, in fact, neonatal outcomes plateaued. We did not see a lot of improvement in outcomes, despite a lot of new technologies and treatments that we were introducing into neonatal intensive care. At the same time, we and many other people around the world noticed that there were a lot of variations in outcomes if you looked at different NICUs. So for example, this is one of our early publications in Canada. And on the left-hand side, you will see variations in neonatal mortality across different NICUs. On the right-hand side, after risk adjustment, you will see there still exists different mortality rates in different NICUs. So these variations, or what we call them small area variations, present an opportunity for us to see how, if some units are doing better, why are they doing better? And whether we can learn from each other to improve our outcomes. Now, we then started looking at, okay, if we have all these outcomes in Canada, how can we improve the quality of care? And traditionally, there are 2 methods by which people use to try and improve and change practices. One is what we call the RCT, or randomized controlled trial, and the other is called continuous quality improvement. Now, RCTs, as you all know, are very scientifically rigorous. However, they are good for evaluation of single interventions, not so good for multifactorial interventions. They are time-consuming, they're expensive, they do not address organizational behavior or culture. It does not address implementation strategies, and the results are often not field-tested. CQI, on the other hand, has been used by hospitals for a long time. However, many scientists consider them to be very subjective. Typically, in an organization, you would have a small group of people who look at what the things that they're not doing so well. They go to the famous hospital down the road, see what they do, and then copy a whole bunch of stuff. And how do you know that this stuff is actually applicable to you? How do you know that it actually works? How do you know that some of them are not even harmful? However, they are suitable for multifactorial interventions. They have a rapid cycle concept, they're relatively inexpensive, they are aimed at changing behavior, which is critical for improving quality of care. They have a strong implementation strength, and the results may be field-tested, but they are sometimes not institution-specific. So both of them have their strengths and weaknesses. Now, when we talked about trying to introduce CQI into our hospitals in Canada, we found a lot of problems because our scientists objected to it. They said this was very unscientific, and that surely we cannot do something like this because it's not scientific enough to be acceptable in our NICUs. And so we were forced to see how we could change that whole thinking. And so we came up with something called EPIQ. Now, the idea behind EPIQ is to try and introduce science, more science, into quality improvement so that it would in fact be acceptable to our scientists and that we could then use it for objectively trying to improve the quality of care. Now, EPIQ is based on 3 pillars. First of all, evidence. We look at the published evidence. We also look at evidence reviews and clinical studies. But more important than that, we also use our own local data that's collected both nationally in Canada as well as within our specific NICUs to help guide the practice changes that we will introduce so that we can target specific practices for intervention at specific hospitals. And I'll show you in a little while how we do this. Second, we look at the context. Because if you just ask people to make changes, they will often tell you, yeah, we know that, but it cannot be done here. And what they are really referring to is a culture within each organization. It's not enough to know what you need to change. It's also important to look at the culture, the behaviors, and how do you actually overcome the barriers to change. Otherwise, you cannot actually successfully introduce change. And thirdly, facilitation. Even when people know what the things they need to do, even when people know what kinds of culture they have, sometimes administrations in individual units find it very difficult to make changes. And we have to facilitate that. So we have a team, for example, that before we even start to introduce any changes, we'll go into a unit, we'll study the culture, the organization, the barriers to change, and we make recommendations to them about what are the things they need to introduce in terms of behavioral and cultural change before they even introduce quality improvement into their units. Secondly, when they start to introduce changes, if they run into trouble, we have a panel of experts that they can consult and a fire team that can fly in, or call them a SWAT team, to help them facilitate those change, intervene with administration, etc., to make those things succeed. So having developed that methodology, we wanted to test it to see whether it would work. So we conducted a cluster randomized controlled trial using this method to reduce nosocomial infection and bronchopulmonary dysplasia in Canadian NICUs, basically to test whether this method will work. We took 12 NICUs in Canada and we divided them into 2 groups. One group we asked to reduce nosocomial infection. The second group we asked to reduce bronchopulmonary dysplasia. There was a third group of 5 NICUs that did nothing, and we used them basically as chronological controls. So here's an example of how we make it work. So first of all, to reduce nosocomial infection, we examine our data to look at what we call the attributable risk. Now that's different from odds ratios, all right? Odds ratios tells you how high the risk is. But attributable risk refers to how much of the problem or variation is actually attributable to that risk. In other words, something may have a very high odds ratio, but in fact only accounts for 5% of variation. Now you're not really interested in that because it means you're going to be spending a lot of time and effort trying to change something that will only affect 5% of your problem. So you want to address those things that account for a large part of the problem, 40, 50 or more percentage of your attributable risk. Now in nosocomial infection, we found that in fact central lines accounted for 40% of all the infections in the NICU, in other words, a very large attributable risk. So this is one of the key things that we targeted. We then looked at different kinds of lines and we found that there were different risks for different kinds of lines, and the catheters that had the most highest risk was in fact the percutaneously inserted long lines. So these were the ones we wanted to target. The problem was that when we looked at the literature for PICC lines, as we call them, the literature is kind of confused. In some of the literature, it says that using PICC lines reduce infection rates. In others, it says it actually increased infection rates. So we looked at our own individual hospital data. Now in this hospital, using PICC lines in the green line reduced the infection rates throughout the duration of use of the lines compared to not using PICC lines in the black line. So using PICC lines in this hospital actually reduced the infection risk. Now in this hospital was quite different. Using PICC lines initially reduced the infection risk, but after a period of time, the infection rates actually went up higher than not using PICC lines. So these guys were pretty good at inserting their lines. They were not very good at maintaining the lines. And therefore, we had to look at their line maintenance protocols to see how we can change those practices. Now this hospital had no clue what they were doing. Using PICC lines did not reduce their infection rates, and then they went up beyond the rates of not using PICC lines. So therefore, their line insertions were very poor, and their line maintenance were very poor. Now this was the most interesting hospital to us, because using PICC lines, reduced the infection rates throughout the duration of use of the PICC lines in the green line. However, the overall infection rate was one of the highest in the country. So this is a dilemma. Because normally in quality improvement, you go to the hospitals that have good outcomes and you do what they do. And you go to the hospitals that have the bad outcomes and you don't do what they do. Now these guys have bad outcomes, but they seem to know what they were doing with the PICC lines. So this was unusual. Now it also explains why EPIQ is different, because we try to be very objective about our data and try to use it to address the problems that we have. So we went to this hospital to try and find out what was going on. What we discovered was that they knew they had a very high infection risk. And therefore, 2 years prior to this study, they actually introduced line management protocols that significantly reduced the infection rates for central lines, and they were successful at doing that. The problem was that they did not address other areas that were also important to infection, such as skin sepsis, skin protocols, etc. And therefore, they continued to have high infection rates, but not because of their central lines. So the solution for us was that we sent all the other hospitals to this hospital to learn how to do central lines, and we sent this hospital to the other hospitals to learn how to do everything else. And so that's why EPIQ is different, because it targets specific interventions at individual hospitals. So we then did this for 2 years, and as you can see the results here, the nosocomial infection group reduced nosocomial infection rates by 34%, which was significant, and there was no change in the BPD rates, which was what we would expect, and so they were good at reducing the nosocomial infection. The BPD group reduced their BPD by 15%, which was also significant, which was good. Much to our surprise, they also reduced nosocomial infection by 44%. Now we don't know why that happened. It could be that this is just a Hawthorne effect. The fact of being in the study actually reduced infection rates. It could also be that in fact these babies did not have BPD, and therefore they could feed, they had less need for TPN lines and so forth, and therefore they had less infection risk. Now the control group had no change in either nosocomial infection or BPD, and therefore what this study shows us is that a method like EPIQ can actually work. In addition, there was an estimated cost savings on reduce of length of stay of approximately 2 days per patient at approximately $7 million per annum. So it could not only improve outcomes, but actually reduce costs. So we said, this is great. Let's take everything that they have done, let's turn them into guidelines, and let's give them to the rest of the country, and let's see what happens. So we watched for another 2 years. The group that was initially asked to reduce nosocomial infection continued to improve. There was another 14% drop in nosocomial infection rates. The group that was asked to reduce BPD had another 8% drop in BPD. So again, there was a continued improvement. So something had happened in these units, so that even beyond the study, they continued to improve and change. Obviously, there was a culture change. What surprised us was that in the control group, nothing happened. There was no reduction in nosocomial infection or BPD, despite us giving them the best practice protocols from those units that had participated in the original study. Now what this tells us is actually consistent with the literature, which says that just giving practice guidelines alone does not work. It is all about behavior. And you actually have to change the culture of people. And therefore, by participating through the whole process that I explained earlier on, there was a change in culture of the initial two groups, the NI group and the BPD group. But just giving guidelines doesn't have the same impact. So we said, okay, that doesn't work. So we started a new study. We got $5 million to start a new study. This time we enrolled all 29 NICUs because 2 of them were merged from 30 to 29. And we enrolled all of them into this study. This was a before-after study because nobody wanted to be any control group anymore. And we targeted all 5 major morbidities as opposed to targeting one at a time. We also decided that we would go around the world and see what kinds of practices we could learn from other people. And we wanted to look also at long-term outcomes at 18 months and 36 months in terms of neurodevelopment. And then to develop a best practices model for Canada and establish a national infrastructure for continuing quality improvement work. Here's the results at the end of 3 years. Nationally in Canada, we have seen a 23% reduction in ROP, a 23% reduction in nosocomial infection, a 20% reduction in necrotizing enterocolitis, and all these are significant results. There was no significant difference in mortality, BPD, or IVH. However, the numbers were actually lower. It's just that they did not reach statistical significance. And I'll explain later on that if you look at the subgroup analysis, the reason was that we actually saw a significant improvement in babies 26 to 28 weeks, but but a smaller reduction in the babies less than 26 weeks, and that actually diluted out the results. There was an 8% increase in survival without major morbidity. And again, I'll show you some of the subgroup analysis later on. So these are good numbers. What it says is that this kind of a method can actually work because this is a significant reduction in morbidity. Now, bear in mind that Canadian outcomes are actually pretty good. We are comparable to most other countries around the world. And therefore, being able to reduce them further after 20 years of no improvement in outcomes is actually a good result. Now, if you look at the adjusted risk ratios, actually the results are very good across the board. ROP down by 60%, nosocomial infection down by about almost 40%, necrotizing enterocolitis by about 30%, and bronchopulmonary dysplasia and IVH were also down, although they were not significant statistically. And survival without major morbidity, again, was down by almost 40%. Now, I said that we then went around the world to see what we can learn from others. And we said, well, that's great. We can improve by looking at ourselves and comparing with each other, but maybe there are people around the world that are doing different things, and there are lessons we can learn from them. So I'm going to describe a few of them for you. I can't do everything, but here's a few examples. First is family integrated care. Now, this is something we learned from Estonia, because Estonia had a very different model of care compared to most other countries. They were part of the Soviet Union originally. They had no money, and therefore they did not have any nurses. And because they were short of nurses, they used parents to look after babies instead. If a baby is admitted to any NICU in Estonia, the mother is also admitted, and she stays there 24/7 to look after the baby. Now, this model of care is actually quite interesting because the parents are the primary caregivers, not the nurse. The parents are responsible for all aspects of the care, except for IV and procedures. They participate in ward rounds, they give medical reports, they do charting, and they participate in the daily medical decision-making of the baby. The nurses, on the other hand, are teachers and consultants. They do not provide care for the babies, unless the mother, for some reason, is called away. Now, they also encourage kangaroo care and developmental care. So this is a very different concept from what most of us practice in the NICUs. And I went back to Canada and I talked to our colleagues to see whether this would work. Well, the doctor said impossible. The nurses said, you're just trying to steal our jobs. Nobody liked it. So I talked to the parents and they said, great idea. So I formed a committee with some doctors, nurses, and parents, and I put the parents in charge. And I said, you tell us, how do we make this work in Canada? I know that Canada is not Estonia, we're a different society. Think about how to make it work in Canada. The doctors and nurses are here to help you and give you advice. They came back to me after 6 months with a protocol that was this thick. I read through it and I was totally impressed. So we did a pilot study. Many nurses were hostile, they refused to participate. So I called for volunteer nurses, and 40 nurses participated. Now this is at Mount Sinai Hospital. Don't forget, Mount Sinai is the largest NICU in Canada. We have over 1,200 admissions a year, and it's the largest acuity, and also the only quaternary center in the country. So here's the results. After our pilot study was published this year, earlier on, after we recruited 40 patients and 80 matched case controls, weight gain in the group cared for by the parents increased by 25% compared to the group looked after by the nurses. Breastfeeding rates doubled. Infection rate in the nurse cared for group was almost 10%, 9.7%. In the parent group, 0. ROP was 14%. In the group cared for by the nurses, 0. In the group cared for by the parents. Critical incident reports, mostly medication errors, decreased by 25% in the parent group. Parental stress decreased, with the parental stress index score decreasing from 3 to 2.3. So overall, it's demonstrated that this kind of a method actually can work in Canada. It's feasible and it's possibly safe. And so, as a result of that, we are now in the middle of a cluster randomized controlled trial at 20 hospitals, with 10 hospitals acting as controls and 10 acting as interventions. We hope to finish this trial next year, and if the results are as good as what we have seen in the pilot study, this will be standard of care in Canada after next year. Now let me talk about necrotizing enterocolitis. Now the problem here is that in most countries around the world, it's around 6% or so. But in Japan, it's 0.5%. So that puzzled us. So I went to Japan to talk to our colleagues, and I said, what do? Do you eat sushi? You know, and they said, we don't know. We do know that 10 years ago, we also had 6 or 7%. But we did a whole bunch of changes, and something worked. And now it's 0.5%. So, not knowing what happened, I sent a group of people to Japan, and they spent a week visiting 3 different hospitals which had very good outcomes. And I said to them, just write down everything they do different than us. So they came back with a long list. And we did literature reviews, systematic reviews, Cochrane reviews on all of these things, and there was no evidence for any of them. So what do? Do we do a clinical trial on each one of them? Impossible. By the time we do all that, I'll be retired. How do we know that some of these things are not acting together synergistically? So that if we do a trial on each one of them, it will fail. But if we do them together, they will succeed. Now that's actually a whole new methodology. Our methodology is actually very reductionist. We try to reduce it to that one thing and test that one thing. How do we know that things don't work together? And how do you use a scientific approach to define that? So I said, okay, we're going to do something different here. Let's see what makes sense, common sense. And the 2 things that struck out at me were, number 1, they use exclusive breast milk feeding in the nursery for babies under 28 weeks. No infant formula is allowed. Now we know that breast milk is good. We don't have to test that. The second thing was they used aggressive feeding. All their babies, even at 22 weeks, were on full enteral feeds with no IVs and no TPN in 7 to 10 days. Now, in Canada, we were very wimpy, shall we say. We were very afraid of NEC, so we took a long time to feed these babies. And I said, maybe it's because we use cow's milk. That's why we have those problems. Maybe if you use breast milk, you can feed them faster. So we took 2 hospitals with the highest incidence of NEC in Canada, and we introduced these 2 regimes, exclusive breast milk feeding as well as aggressive feeding. Within 1 year, we got them down to about 1% in both hospitals. With this data, I went to the government and we asked to build milk banks for donor milk across the country, and we started doing that. And I can tell you that in Ontario, for example, we have now a milk bank that opened up about a year ago now, and we are supplying milk to all the babies in Ontario. And starting in Toronto, I can tell you that in Toronto, with the 3 NICUs, in the last year, we have seen a 2/3 reduction in necrotizing enterocolitis, just in 1 year. So this is pretty dramatic in terms of the outcomes. I know some of my colleagues tell me they're also using breast milk, they don't see this reduction. And maybe the perinatal risks are different, I don't know. But certainly in Canada, we are seeing a huge reduction in NEC rates just by doing that. And now we are opening milk banks all across the country. And by next year, I hope to have enough milk for every baby under 28 weeks in Canada for breast milk exclusively. And I intend to ban cow's milk from all these babies. And if the mothers want them, they're going to have to give informed consent to get cow's milk. So, with those results, we said, okay, we've done well so far. Now, where do we go with next? Next year, we're starting EPIQ-III. And our objective is to drive for 0. 0 morbidity, 0 mortality. Now, we're not going to get there, we know that. But we're going to try and get much further down than we have done so far. How are we going to do this? First, we're going to use more refined data analysis, and I'll show you in a minute how we do that, to target specific outcomes for intervention. We're going to audit interventions and impact so that we can understand more clearly which are the things that work or don't work. We're going to use an accountability framework so that people are going to have to account for why they're not doing things that are important or why they're not achieving results. We're going to target not just outcomes, but also processes of care, because these are very important in terms of actually improving outcomes. We're also going to start to look at the systems of care, because it's not just important to deliver clinical care at the bedside, it's important to look at your processes of care and the systems of care that support that. All 3 of those are critically important. And therefore, we're going to start to look at processes like transport, antibiotic use, patient safety, and so forth. So here's an example of the things that we're going to target. I mentioned earlier on that in EPIQ-II, we found that most of the gains were in fact in the 26 to 28 week gestation. For example, there was a 17% increase in survival without morbidity in that group. However, in the babies less than 26 weeks, we did not see much improvement. we saw only a 3% improvement in survival without major morbidity. And certainly in the 26 to 28-week group, there was a significant improvement in IVH and BPD, whereas in less than 26-week group, we did not see that. Therefore, one of our strategies is going to have to target babies less than 26 weeks, including how do we resuscitate and stabilize these babies, the golden hour, ventilation, fluids, nutrition, and so forth. I think we can make a significant dent in this group. We're also going to look at hospital-specific targets. Here, for example, is one of the maps that we create for our hospitals. And the color codes allow hospitals to look at which are the areas that they should be concentrating on, because that's where they need to be focusing based on their results. We're going to look at specific practice targets. So, for example, IVH, BPD, etc. We've been analyzing the different interventions that people use in different hospitals and looking at which are the ones that really had an impact in terms of reducing the risk. So here, for example, is efforts that have been done by some of the hospitals to look at trying to reduce intraventricular hemorrhage or neuroprotection. And you can see that there was not much impact from delayed cord clamping or magnesium sulfate. On the other hand, not using bolus fluids and minimizing the use of inotropes resulted in a significant reduction in intraventricular hemorrhage. So analysis like these start to give us clues about the kinds of things that we need to be targeting, because those are the ones that probably will have the impact in terms of reducing morbidity and mortality. And we do this for all the different outcomes that we try to address. Now, all these kinds of efforts will have huge impacts on the healthcare system. For example, we're building milk banks across the country. If parents provide care, we're going to have to relook at all the different kinds of personnel that we use. We have to redevelop our facilities. Now, there's a trend nowadays to look at single-room design for NICUs. But if you really want to have parents providing care, is that the right design? Because we find that, in fact, the parents help each other. They teach each other. 50% of the teaching in the NICU is now done by parents. They support each other. And does single-room design prevent that? More appropriate use of technology, resuscitation methodologies, will allow us to reorganize our regionalization of care and change the way in which we transfer patients. It will also change the roles of all our staff, from nurses to doctors, etc. Now, these are huge changes in the healthcare system. It also means that we can reallocate our resources and potentially get better outcomes at reduced costs. Now I'm going to talk about some of the considerations for improving care that we need to be thinking about going forward. First of all, we need to start moving away from just looking at evidence research to implementation science. Many of us are not trained in implementation science. It's a whole different ballgame. It's about psychology, it's about behavior, it's about program evaluation, it's about program management, it's about systems change. And we need to be thinking about these things if we're actually going to improve the quality of care. Second, we need to embrace new research methods. Methods that involve mixed methodologies, pragmatic trials, etc. Because RCTs cannot do everything. Yes, RCTs are important for certain things, but for some things we need to use different kinds of methodologies and to develop them so that we can actually use them in our work. Third, we need to reconsider our approach to evidence, because if we're going to embrace these other research methods, how do we use this evidence that we're going to be developing? And how do we ensure that we can appropriately use them? We need to move our approaches from just looking at improving outcomes to actually improving processes of care and systems of care, because those are equally important in terms of determining outcomes. We need to consider not just the odds ratios, but the attributable risks, because if you actually want to make a difference in terms of improving outcomes, you have to look at what really matters in terms of changing those risks. We call them attributable risks. We need to be more family-centered. I believe that modern medicine, we have moved too far away from the family, and we forget how important that really is. That piece of family-centered care is actually really important. And it's not just the technological and pharmacological aspects of care. Family-centered care actually has a lot of benefits besides psychological benefits, and I've told you about that earlier on. We need to integrate research, clinical care, and systems management in collaborative networks. So in our network, for example, we do everything in the network. It's not just looking at outcomes improvement through using quality improvement. We also do clinical trials. We do everything through there. Depending on the methodology that's required for a particular objective, we utilize that methodology. So we don't create separate networks for everything. We need to adopt a principle of accountability. so that if people are not producing results, they need to account for why, and they need to do better than what they are doing. We need to rethink the reductionist approach. I believe that it's a mistake to just use the reductionist approach, because there are many things that have to be done not in sequence or not individually, but together, and that's where you get the best results. And finally, I think we need to be humble and to be open to new ideas. For example, when I talk about Estonia, about Japan, none of them are evidence-based places. Yet, why do they produce such good results? I think it's not that evidence-based medicine is wrong. What it means is that we have to be open to new ideas, we have to come up with innovative new ideas, and then we have to test them and see whether they will actually work. Now, the EPIQ system is now being adopted by many other countries, including in Sweden, which is going to start next year. We have an annual conference, which is attended now by nearly 200 people from many countries around the world, and it's a roll up your sleeves conference. We get together, we roll up our sleeves, and we don't have guest speakers to come and talk about different things. Rather, we discuss what are the things that work or don't work, new strategies, new ideas, and what we're going to do for the next year. Thank you.